To understand the law of independent assortment, let's consider one of the dihybrid crosses conducted by Gregor Johann Mendel. In this cross, Mendel mated a pea plant having round yellow seeds with a wrinkled green-seeded pea plant. All F1 plants had round and yellow seeds. He then allowed F1 round and yellow-seeded plants to self-fertilize and produce F2 progeny. Can you guess the types of gametes formed and the possible genotypes and phenotypes of the F2 progenies? Mendel found that the F2 progeny plants were produced in a phenotypic ratio of 9:3:3:1, with 9 round and yellow, 3 round and green, 3 wrinkled and yellow, and 1 wrinkled and green. Mendel found a similar ratio of 9:3:3:1 for different combinations of the 7 traits that he selected for these breeding experiments. On the basis of this dihybrid ratio, Mendel proposed a generalization, which is often referred to as Mendel's law of independent assortment. This law states that when 2 or more pairs of characteristics are combined in a hybrid, each pair separates independent of the others during gamete formation. In other words, characteristics are inherited independently of each other. In our example, the genes for seed color and seed shape are sorted independently, resulting in formation of 4 types of gametes in equal proportion. We can examine these 2 characteristics individually in the F2 progenies. First, seed shape. There are 12 round and 4 wrinkled seeds, representing a ratio of 3:1. Similarly, there are also 12 yellow seeds and 4 green, again showing a ratio of 3:1. So regardless of the color of the seeds, the ratio of the shape of the seeds will always be 3:1. Similarly, regardless of shape, the ratio for color of the seeds will always be 3:1. In the heterozygous parent plant, the alleles for seed shape, capital R and small r, and alleles for seed color, capital Y and small y, assort independently. 4 types of gametes are produced in equal proportion. These are capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. As you can observe in the Punnett square, the fusion of these gametes produces the F2 progeny plants in the phenotypic ratio of 9:3:3:1. Gametes are formed as a result of meiosis, a type of cell division. The answer to independent assortment of the genes lies in the behavior of chromosomes during gamete formation. Consider this parent cell with 2 pairs of homologous chromosomes. Although the homologues look similar, they have different alleles. One has the capital R allele and the other has the small r allele for the seed shape. Similarly, the other homologous pair has the capital Y and the small y allele for the seed color. Before meiosis, the chromosomes replicate and each chromosome carries a pair of identical alleles. During meiosis, the homologous chromosomes pair up and align in the middle of the cell. There are two alignments possible for the homologous chromosomes. In first case, the two dominant alleles, capital R and capital Y, are aligned together, whereas in case 2, they are aligned opposite to each other. As the chromosomes move to the opposite poles, the alleles segregate. Notice that in the first case, capital R and capital Y alleles are in the same daughter cell. In the second case, the two dominant alleles are in different daughter cells. Notice that the alleles for seed shape have segregated independently of alleles for seed color. This is the basis for Mendel's law of independent assortment. In the second phase of meiosis, the cells divide again to form gametes. Notice that there are 4 different types of gametes. Are 1/4 formed with capital R and capital Y, 1/4 with capital R and small y, 1/4 with small r and capital Y, and 1/4 with small r and small y. Mendel's law of independent assortment is not universal. It holds true for those characteristics where the genes are located on different chromosomes. It is important to note that genes located on the same chromosome may not assort independently. Such genes are said to be linked.